Hi everyone, my name is Dirk Faber. I am the teacher and course coordinator of Light Tissue Interaction 2020. Unfortunately, everything is still different this year, so we cannot meet in person for class. I still hope we can make the best of it, and I would like to invite you now to watch the attached video where I will try to discuss some household settings for the coming course. And I hope to see you all in good health next Tuesday, tomorrow. So unfortunately this year everything is still different. So therefore I would like to use this segment to talk about the goals of the Light Tissue Interaction course, the format of the classes and the examination, the tools that we'll use along the way, also about the schedule and the teachers, and finally an introduction about myself. Why am I working in this field? And give you some contact information. One of the main goals of this course is to give you a broad overview of medical imaging and diagnosis based on the interaction of light with tissue. I'm going to make a little bit of a disclaimer here. I tend to use the terms light tissue interaction and biomedical optics and maybe even biomedical photonics a little bit interchangeably, but be assured that they mean the same thing. And apart from the overview, I would like to combine that by discussing an in-depth treatment of state-of-the-art biomedical optical techniques. Before we can do that, we need to get ourselves acquainted with optical properties and optical theories, such as Rayleigh scattering and Mie scattering. We need to discuss the main interactions of light with tissues, such as absorption and scattering, and look at important tissue scatterers and absorbers, and see how their optical properties may change when we change their densities. For example, if we add more molecules or keep adding more molecules, how does the absorption and scattering properties change? We need to study electromagnetic wave propagation in tissue, in random media, tissue-like random media. Also, I would like to compare the performance of different techniques with different settings or different configurations, or compare different techniques that are applied to the same medical problem. So let's say imaging the same tissue. And finally, it would be great to translate these fundamental physical concepts into really practical clinical applications that a doctor could use. All right, let's talk about the schedule, the format of the classes. We have been given 2 sessions per week of about 4 hours each. And I think it is in the best interest of all of us that I am not spending 4 hours talking. So I think it will look something like this. Maybe it can change a little bit during the course of the course. An introduction on a topic where I talk about, let's say, 45 minutes about a certain topic. Then I'll give you an assignment to work on alone or in small groups for about 45 minutes. A discussion of that assignment, maybe some additional materials for 45 minutes, a new assignment or a continuation of the previous assignment, also for that amount of time, and then we'll end the session with a discussion and an assignment of some out-of-class work. Of course, my goal is to be available during assignment time for these central questions and clarifications. I guess it will depend a little bit on how the course goes and the mastery of the tools that we have available, how this will look eventually. And then, of course, another topic that is close to our heart is the assessment of the course, the examination. According to the study guide, the course is evaluated using a written open book exam. Given the circumstances, that is not possible. So this year, the grade is composed of 3 written assignments, which you can make by yourself or in a small group. And then in the end, there will be an individual oral exam at the end of the course, which will take about 20 minutes or so. Details of that we can discuss later on, but for the oral exam, at least because that is potentially the only individual examination that we will take. And 6 or higher, a pass or higher is required. Of course, since everything has to be online, we should decide on a platform to use. I have had some experience with Google Meet, which was good for me, but I think Zoom may offer a little bit more opportunities. Even though it is only one day before the class starts, I have not yet decided. But I will invite you to a meeting on either platform via an announcement on Canvas to make sure this happens. Of course, Canvas will also be used to exchange materials, to exchange messages via announcements, and I will make materials available as much as possible before each class and send notifications through that. I will also set up a YouTube channel for which you will also get a link because experience learned that eventually Canvas will run out of space to store all the recorded lectures. Regarding the schedules and the teachers, for this course, for the first time, actually the first 3 weeks are shared with the biomedical optics course, given at the VU, from which I'm also the coordinator. There is some shared basic knowledge between the two courses, so we decided that it would be opportune to share the first 3 weeks of the courses. So that means that this week we'll be talking about absorption and scattering together. Next week about the fusion theory Monte Carlo methods, then optical coherence tomography, and after that we will go our separate ways, where the biomedical optics course will focus more on clinical applications and applications on sich, and we in the LTI course will focus more on the really physics foundations of the interaction of light with tissue. So that's it. The schedule for LTE is not quite yet finished. So when I have that, I will update you, of course, as soon as possible. Your lectures in this course will be me and Professor Janus de Boer from the VU, who will talk about OCT and about Raman scattering. So then I would like to give you some contact information. Of course, I will be on Google Meet or Zoom or whatever platform we settle to during class hours. I will be either talking or I will be available. And then for non-urgent matters, you can send an email via Canvas or directly to my email address that is given here. And of course, I'll respond as soon as possible. For the really urgent matters, so let's say you cannot get the connection to work when the class starts, then you can also send me a WhatsApp at the number that is listed here. So now I would like to use the remainder of this presentation of this segment to introduce myself to you and illustrate to you why I am in this field of biomedical optics. I have been working on optical coherence tomography for about 20 years now in the Academic Medical Center of the University of Amsterdam, where OCT, optical coherence tomography, is kind of like the optical equivalent of ultrasound imaging. So on the left-hand side of this slide, you see a familiar picture of a baby in a womb, where the grayscale represents reflectivity versus depth. With OCT, you can do more or less the same, only not using sound waves, but light waves as the probing mechanism, which means that because of the lower wavelength, you can look at much smaller details. So to illustrate here, for example, it would mean that if you would zoom in on the hand of the baby in the left-hand picture, you would get the wildly spinning picture on the right-hand side, which would look into the skin of that baby using OCT. You can recognize here a spiral structure that represents a sweat gland in the OCT image. So over the years, OCT has become a standard method in ophthalmology. So if nowadays, and I hope you will not be in that position. But if you have to go to an ophthalmologist in an academic medical center, then the chances are high that you will get an OCT scan of your retina, of the netvlies in Dutch. So I take an echogram of the retina for diagnostics. So, for example, what you see in this picture already from a couple of years ago from one of my PhD students, Roddy Kinkeler. On the right-hand side, you see a conventional so-called fundus image, where a photograph is taken of the retina, and you can see some blood vessels over there. You can see, but you can also see some anelomes, the yellow dots. Based on this image alone, it is hard for the ophthalmologist to make a diagnosis, but given the 3D information that OCT provides, it is easier, perhaps clearer, to actually make a diagnosis. But we were not entirely satisfied with this. We had the feeling that there was more information in these OCT images than just representing 3D pictures. So one example you see here in the graph over here, if we can increase the resolution of OCT by, for example, using adaptive optics, which is a very neat technique where you change the focus of light using some clever techniques, you can actually increase the resolution down to the scale where individual rods and cones can be visualized within the retina. And not only visualized, it is also possible to actually tag them and follow them over a period of time. So that means that quantification of these rods and cones of cell layers can be followed and then can be associated with the development of retinal disease. Of course, it also means that you can do a lot of post-processing on these images. You can, by increasing the resolution, so here you see a picture of the retina where from left to right the resolution, the optical resolution is basically doubled, the sampling resolution is basically doubled. You can see that a lot more information can be obtained. And also that it becomes more feasible and more easy to automatically segment layers in these samples. So all in all, we have seen that OCT has become a fixture in ophthalmologic research, and I was particularly proud of this image on the website of Biomedical Technology and Physics, where one of our previous students, Joy, is depicted doing OCT research here. So after that, we were not entirely satisfied, in part because there were so many people working in ophthalmology, and you have to make your point at in some way. So we decided to look if OCT could be a good technique to distinguish different tissue types in another field. One of our first endeavors was in cardiology, where we tried to differentiate between calcified tissue and lipid tissue within a vessel wall. You can see here cross-sectional images of a vessel wall with a calcified lesion indicated by C and a lipid lesion indicated by L over here. And as you can see, also there is not a very clear distinction based on the image alone. So what we decided to do was to look at not at the grayscale image itself, but look at the signal versus depth within such an image. So we selected a region which we thought which was calcified, or we thought was a lipid region. Plotted the signal versus depth, and then looked at how strongly the signal decays as a function of depth. We can quantify that into a coefficient, here labeled mu t, the attenuation coefficient, which has then units of decay per millimeter, as you can see. And it turns out that this can actually be used to differentiate between these different tissue types. We can plot different regions within the vessel wall on a color scale from high to low attenuation coefficient, and we can see that this was actually able to differentiate between these two tissue types. This was somewhat successful. We moved on to see if we could actually distinguish between different types of tissue in cancerous tissue. In panel A in the figure, you can see a 3D rendering of a piece of tissue that is suspected for cancer. In panel B, you see that same piece of tissue, but then sliced up in different cross-sectional images. In panel C, from each of these cross-sectional images, we can plot the OCT signal or the reflectivity as a function of depth. And from these, we can see different regions which correspond to different tissue layers. And we can actually measure how fast the light attenuates in these different layers. And that is our metric. That is what we intend to use to differentiate between different tissue types. And that is what you can see in panel D. If we, for example, look at actually the slope of this red curve in panel C, so the degree of decay in signal attenuation, we can see that there is a difference between healthy tissue in blue and precancerous tissue in purple in this case. So we can actually use this metric to discriminate between tissues that are cancerous and that are not cancerous and that are precancerous. So this was a very long introduction to actually bring me to my motivation for this work, because I want to know how do these changes in tissue functionality, into tissue morphology during the development of cancer or during the development of disease, how do these actually change into optical properties that we can apparently measure? How does that mechanism work? The second question is, how can I measure these optical properties, but I want to measure them quantitatively. I want to make sure that if I measure with one technique, I measure the same thing with another technique. I want to make sure that if I measure it if I measure it now and I measure it in 5 minutes or so, I want to measure the same thing. And then the last question, of course, is how can I translate this optical measurement that I just made into a meaningful clinical diagnostic, something that the doctor can actually use to make a diagnosis. Of course, we have some ideas. The cartoons underneath here illustrate the changes that cells undergo as cancer is developed. So let's focus on the right-hand side first, where we have normal cells with let's say normal nuclei and normal cytoplasms. If cancer develops, then the nuclei get more irregular, the cells themselves get more irregular, the ratio of nuclei and cytoplasm changes. So there is a lot of very small scale morphologically going on that could actually change the optical properties, so that we could actually measure. And if you then look at microscopic slides, and that is then the images on the left-hand side, you can see for example, for normal tissue, you can see indeed a very regular distribution of nuclei, more or less equally sized nuclei. And then if you then look to the renal cell carcinoma on the right hand, the malignant cells, you see that the distribution of nuclei becomes much more irregular. You can see that the distribution of sizes of the nuclei becomes much more irregular. So there is a lot of small scale morphological changes going on that will surely translate into changes in optical properties. So that will surely translate into something that we should be able to measure with optical techniques. But the big question for me is how can we make this quantitatively? How can we understand how these changes actually work and how these changes actually translate into changes in optical properties? That's it for now for me. We have a lot to discuss, of course, so I hope to see you on Tuesday, and in the meantime, stay safe. See you tomorrow, I hope.